Hi everyone, myself Dr. Manohar, working as a professor in VGS College of Pharmacy. So today I'm going to discuss about this diagnosis of the COVID-19 or SARS-CoV-2. So here in this I'm going to discuss about the basics about the corona and protective equipments for the clinicians and technicians, pathophysiology of the coronavirus, and diagnosis, various techniques like RT-PCR, antibodies, detection of antibodies, and rapid antibody-based blood test. So coming to the COVID-19, it's an RNA virus. It is spread by the droplets. COVID-19 has 500 micrometer in size and cannot travel more than 1 meter distance while we'll talk. So that is the reason we have to maintain the social distance at least 1 meter. Virus can spread through the biological fluid, unhygienic behavior, and even the fecal contamination. Virus may present more than 3 hours after sneezing or coughing by the patient or individual. The virus can infect up to 24 hours present on non-living surface. So any surfaces like normally will touch, normally the lift or whatever lift button or any surfaces will touch normally. So the virus may present up to 20 hours. So here the protective equipments for the clinicians and technicians. The one who is dealing with this COVID patients should has the N95 respiratory mask, gloves, gown, and protective glasses. So here normally we'll collect the sample by the swab. So calcium alginate swabs should not be used, or wooden shafts must not be used because it may damage your, and calcium alginate can inactivate the virus. So yeah, so here normal the synthetic cotton swabs are going to be used by using nasopharyngeal or oropharyngeal route. Gently touch the surface and immediately shift these surface to the like viral in a transport medium. So before going to diagnosis, we are going to discuss about this pathophysiology of the corona. So corona, as we discussed, it is a RNA virus, so it can directly fuse because the outer layer of the corona is made up of a lipoprotein, lipid membrane. So here for the entry of the virus into the surface, here ACE2 receptor is helpful. So ACE receptor is going to be internalize this virus and form the endosomes. So these endosomes are digested with proteolytic enzymes which are present in the lysosomes, and a nascent RNA virus is going to be released. This RNA virus, by using the host ribosomes, is going to be prepared prepared a particular protein called as RNA dependent RNA polymerase, which multiplies the RNA into several folds. So these several folds of the RNA is going to be proteolysis, and convert into various structural and non-structural proteins of the virus. So these are by using the Golgi apparatus. So these are going to be arranged properly, and a number of viruses are going to be synthesized. So this is the so here this arrow mark indicates various structural and non-structural proteins synthesized by the, and the assembly of the virions and the number of virions are going to be released. So coming to diagnosis, so normally we'll use this RT-PCR, the swab which are there in the viral transport medium, okay, are carefully isolated the RNA from these samples. So RNA after isolation, it is very sensitive. So if the normal temperature is going to be degraded, so in that case, so you need to maintain a proper temperature. So around 4 degrees centigrade for proper proper storage of the RNA. So RNA is immediately converted into the CNA, I mean cDNA by a specific enzyme called as reverse transcriptase. So which maintains which conditions needed is 42 degrees centigrade for 30 minutes. Okay, once it is converted to the cDNA, it is going to be stable. So we can go for using tag DNA polymerase and primers, DNTPs, buffers, etc. by say normal simple PCR reaction. Okay, after completion of different 32 cycles around, so these are going to be loaded on this gel agarose, where based on the size of the DNA, we are going to confirm whether the patient is positive or negative. So one more normal test for the viral detection is uh, by uh, by finding a specific antibodies present in this against a particular virus. In case of coronavirus, so we are not recognized a particular epitope which is having specific identification. So here normally when the virus is enters into our body, so the two immunities, cell mediated immunity and humoral immunity are going to be get activated. So this cell mediated first the macrophages are going to be engulfed, and the proteins are going to be present to the T cells and B cells. T cells further activate the cytotoxic T cells or CD8 T cells and memory cells, CD4 T cells, and through macrophages the B cells are going to be get activated and is going to be converted to plasma cells and secrete a specific antibodies which can target to this particular coronavirus. So here in this case first the in ELISA first blue colored one blue colored antibody is the specific antibody which is present antibody which is target to the virus. So it is the commercially ligated on a plastic surface to which the blood or sample is going to be added, in which if the sample contain the virus is going to be attached to this specific antibody and the various antibodies present in the individual. So after washing we'll go for the secondary. We not yet gone to this ELISA because we don't know the specific epitopes which is present on this particular virus. Epitopes is the protein substance which is having highly immunogenic activity, and should be present in all different genotypes of the virus. So here the virus which are present in the China, US and India is completely it is different. Then China it is said to be the very potent virus, whereas India it is less potent virus. And I mean to say the epitopes are going to be changed because depending on the genotype. Genotype is nothing but the specific sequence present on the virus is called as the genotype. So depending on that we need to design the ELISA. So coming to the another rapid antibody based blood test, which is said to be, it is very important to screen the population. So here the problem with the corona is it is not a symptom. All the symptoms are not seen in all the infected individuals. So most of the individuals are asymptomatic carrier. So to trace these individuals and to find out whether the patient is a carrier or not, this is very important to detect the rapid antibody based blood test. So basically the entire population can be divided into 4 groups, both IgG and IgM negative. Second one is IgG only positive, and both IgG and IgM positive, and IgM only positive. So here we need to understand that these are the immunoglobulins. When any pathogen enters into our body, okay, these are going to be prepared. So when the first immunoglobulin which binds to antigen is IgG antibody, whereas it is a simple monomeric immunoglobulin, whereas IgG is a pentameric. So upon the chronic infection, these IgM antibodies are going to be developed. So coming to this, if the individual has both IgG and IgM negative, so the left side you can see this C indicates the control, So I mean to say the control in the sense the strip is working properly or not is going to be indicated by this C. And if the IgG present, so here there is a band in G on G or IgM. So in first case, both IgG and IgM negative. So that indicates that the individual never had the virus. So advantage of this, safe for the society. The individual is safe for the society. Chances of infection minimal unless comes in contact with a positive case. So he's a not infectious, fit to work. So follow up, the suitable to work in safe environment. No treatment is needed for the individuals whose IgG and IgM is negative. So next category, IgG positive individuals. It indicates the asymptomatic carrier, subclinical cases, developed immunity. So advantage, it's a safer society. Chance of reinfection is minimal, and the recovery phase and will improve fast. So most suitable for the corona warriors at a community level. Less need for the intensive treatments. So these are the individual who are fit to treat the corona patient. Maybe he may be police, he may be doctor, he may be nurse, he may be a sister, whatever. So here the individuals who got the immunity against these IgG positive, so they are said to be the corona warriors. At the third case, both IgG and IgM positive. It indicates the asymptomatic carrier, subclinical cases did not develop symptoms, probably will not convert into the clinical case. Early recovery will develop a less severe disease. So here the stage is, I mean, the chronicity is going to be increased in both IgG and IgM is positive. So for this, the individual should be provided home isolation, need conservative symptomatic treatment. Next, IgM only positive. So it indicates the asymptomatic carrier, subclinical cases, late incubation period cases. These are the late incubation period cases. So very dangerous in other terms. So advantages, hidden exposed case, early detection, so hotspot case detection. So here for these individuals, a strict follow-up and isolation is required. These are going to be confirmed by using RT-PCR, and a proper treatment should be given for these individuals to combat with this. Okay, hope you understand this. Thank you. Stay home, stay safe.